Okay, so thanks for joining us. This is a review of DNA replication, RNA, and protein synthesis. Again, please keep your microphones muted so that there is a limited background noise for the recording process. For those who can't join us live, they can watch it later. Okay, so let's talk about DNA and why DNA is important. Make this a little bit bigger. Okay, now is it gonna let me roll through my slides while they're full screen? Yes, it is. Okay, great. So lovely picture of my family, my two cute little boys and my husband from, this is like good 2 or 3 years ago. My little guy's kind of really young there, he's 4 now. When we were in Disney riding Dumbo, we're about to ride Dumbo. And everyone says my two boys look a lot like my husband. They say they look a lot like him, which is fine. I think my husband's a handsome guy, so I'm happy to hear that. But they are mine, I swear. Why? We look like our parents and people who are related to us because of our DNA commonalities. We share more DNA in common with our family members than we do with strangers. And of course, that is going to be more in common in terms of building the structural and functional proteins of our cells, more in common with those proteins than with strangers. So the foundation of understanding what DNA does is to understand this concept called the central dogma of molecular biology. The central dogma states that the molecule DNA is going to code for a molecule called RNA, messenger RNA specifically, and that molecule is going to help build proteins. There are going to be several types of RNA that are involved with protein synthesis, and there's multiple steps involved, but this is the overview. DNA makes RNA, RNA makes codes for building of proteins. Okay, and then the protein's actually built at the ribosome of the cell, just like we learned all the way back in like October when we did cell parts. Okay, so let's talk about this idea. So DNA is going to get transcribed. It's going to undergo this process called transcription in the nucleus. And that process is going to allow the production of this other molecule called mRNA, messenger RNA. This is kind of like DNA's cousin. It's also a nucleic acid. It's going to have a lot in common with DNA, but it's going to have some key differences too, and we're going to highlight those in just a few minutes. And then this mRNA is able to leave the nucleus after being made in DNA's image. It's able to leave the nucleus, and it's going to move out into the cytoplasm to where the proteins get made at the ribosome. And the process by which this mRNA gets read by a ribosome is called translation. So we have two main steps involved with making a protein: transcription and translation. And I did assign you an Amoeba Sisters that highlighted these two steps. Hopefully you got to do it already. If you haven't gotten to do it yet, it'll just make a little more sense as you do it after this lesson. But transcription, translation, two key steps in ultimately building a protein, which is the end goal of protein synthesis, building of a protein. So what do we need to know? First, you got to make sure you understand DNA structure, recognize it in either of its forms. This model here on the left is an untwisted ladder where we can actually see individual nucleotides. Please recall that a nucleotide is a phosphate, a sugar, and a base. You should know the name of that sugar is deoxyribose. You should know that the name of the 4 bases that it could contain in a nucleotide are going to be C for cytosine, A for adenine, T for thymine, or G for guanine. Those are the 4 bases you can find in the nucleotides of DNA. There's going to be a little bit of a difference when we talk about the nucleotides of DNA's helper called RNA. A little bit of structural differences there. We do know DNA is typically twisted up. It's a twisted ladder. We call this double helix, like the picture on the right-hand side is showing it. So please be able to recognize it either way. The picture on the right is not showing the details of the sugar phosphate side chain. We also sometimes refer to these spirals as the backbone of DNA. You don't always see them in the drawing. Sometimes you only see the bases being highlighted like this picture is showing, but it's still DNA nonetheless. Nucleotides, recognize there's going to be a phosphate, there's going to be a sugar, there's going to be a base. Even when we get to talking about RNA nucleotides, still the same 3 parts: phosphate, sugar, and a base. Just a few key differences in the name of the sugar and the name of one of the bases. Please remember that DNA is really, really tightly wound up and coiled in the form of a chromosome. And that chromosome, you have many of them, 46 total, are going to be found in the nucleus of your cell. Okay, so a little scale purposing here. In a cell is a nucleus. Within the nucleus is a chromosome. Within the chromosome is DNA. And if you examine one piece of DNA, one segment of DNA, that's called a gene. And a gene is the section that is a set of instructions that codes for building a specific protein. right? This view of DNA is just showing you the bonding between the base pairs. So you're not going to have to memorize which bases are purines and which ones are pyrimidines. To be honest, the living environment regions level doesn't go into that much detail. But we do have purines and we do have pyrimidines, and that has to do with their size, and that's why we have to have the pairing rule be the rule that we know. And that rule, of course, that we know is adenine pairs with thymine, guanine pairs with cytosine. So A with T, G with C. That's the rule that you hopefully know by now. We did that before we got closed down. So DNA is really, really tightly coiled, wrapped up, twisted molecule stored in this chromatin form when the cell is not dividing, like you see pictured here on the left. But when the cell is dividing, we actually see DNA condense even further into this X-like chromosome structure. Now, we do have 23 pair of chromosomes, and we do have one chromosome titled the X chromosome, but they all have a similar shape to this one, whether they're the X chromosome or not, when they are getting ready for division or undergoing division. So let's talk a little bit more about replication. DNA, if you look at it as a ladder when it's untwisted, that ladder can be broken. And that's what you see happening here by this orange blob here. This is an enzyme. This enzyme is able to basically run up the middle of the ladder and unzip it, just like very much like when you unzip your zipper on your jacket. So this enzyme is going to help with replication, because when DNA has to copy itself, it's got to open up those bases. And it's also going to help out later when we have to open up the DNA for other purposes. When we have to provide instructions for building a protein. So DNA is going to unzip itself for two purposes. It's going to unzip itself when it has to replicate, which is pretty much all the time. If you're ever making a new cell, you got to replicate your DNA first. We learned that when I had my very high-tech models of pipe cleaners. I had one pipe cleaner, became two pipe cleaners, and we said that's the magical process of DNA replication. That's what we're talking about in a little more detail now. How does DNA replication happen? So if you see the picture here on my right, one single strand of DNA, that's actually a double-stranded molecule, is able to split open, and when it's split open, each half serves as what we call a template for a new strand to form. So the white strand is the old piece of DNA, and then this black strand that's forming is the new piece of DNA that's being synthesized during this process of replication. So what's happening is all your free-floating nucleotides that you have floating around in your cytoplasm from breaking down previous DNA, and believe it or not, even from just consuming food, you're eating an apple's DNA when you eat the apple, and then those building blocks get broken down and get sent to your cells. So you've got all these little free nucleotides floating around, ready for use. So when the enzyme broke open those bonds between what was this T and its previous pair of an A, it's giving a chance for a new nucleotide with an A on it, that's adenine, to come in and pair up. So that process happens on both the left side and the right side of that original strand, and ends up letting a new strand form on each side. So that enzyme is called helicase. Think of it like helix. It's going to be breaking the helix. It's unzipping and unwinding the helix. Breaks those hydrogen bonds between the bases, and you end up with the replication fork. That's just this area right here where that DNA was once all together, but now it's being separated. Okay, so at the replication fork is where we have now free-floating nucleotides that are in your cytoplasm from digestion and from breaking down old DNA. We reuse our parts. We don't get rid of things. We recycle here in our cells. And they're just going to come in and attach by biochemical bonding to whatever their pair is. So if there is a now available G, then a free-floating C is going to come on in and attach to it. I'm going to show you this in an animation a little bit more later. So they bond to their complementary pair, and they get attached by another enzyme. And that enzyme is going to be called DNA polymerase. To polymerize means to build, a building molecule, building a polymer, a big molecule. And DNA is, of course, a polymer made of millions of bases long. So DNA polymerase is going to be coming in and helping attach these new black-sided here, in this picture, these new nucleotides are getting attached by the enzyme DNA polymerase. And it really is a little more complicated. There's more than one kind of DNA polymerase. We're not going to get into all of the different number of DNA polymerases, but the general function of DNA polymerase is to help build DNA. during the process of replication. So in the end, once it's done, you should end up with 2 pieces of DNA, whereas you only had 1 to begin with. So you should end up with 2 identical double-stranded DNA molecules, and each one, as you can see here in the picture, each one is going to have half of its strand be from the old molecule and half of its strand be from the new molecule. And so that's why you see the black strand here and the white strand here, and then same thing on the right side, 1 black strand, 1 white strand. And this concept, this model of DNA replication, is called semi-conservative modeling, meaning that semi, or part of the new DNA molecule, has half of the old strand in it and half of a new strand. It's semi-conservative. So a little more detail into how this process works, and one of the videos I sent you may have gone into more detail of like this picture does, is that this process happens by those enzymes that we mentioned, DNA polymerase and helicase, right? So helicase in this picture is the blue triangle. The blue triangle's job is to unzip, okay? Just splitting those hydrogen bonds between those base pairs. It's just gonna keep running along this part of the DNA and keep unzipping it. As helicase unzips, then there's the enzyme DNA polymerase, and DNA polymerase in this picture is represented by this like orangish color rectangle over here and over here. Now, DNA polymerase doesn't know exactly where to go to start building without a little bit of help from another enzyme. And so one of your videos did go into that detail. I'm gonna just show you where it's being represented here. That enzyme is DNA primase. A primer is like a starting point. So if you're getting primed to do something, it's like you're getting prepped to do it. So think of DNA primase as a primer or starting point that tells polymerase, hey, here's where we need to start building. It tells it where to start laying down the new sequences. Now, what happens with polymerase is it only, because of its shape, its biochemical structure, it can only work in one direction. And if you've ever noticed on a strand of DNA, like one of those simple first diagrams I showed you, it looked like the sugars of one DNA strand were pointing up, and the sugars from another DNA strand opposite of it were like pointing down. So if you look at the point of the sugars, the little pentagons in the nucleotides, they're pointing in opposite directions. That's not a mistake in the drawing. That's actually how the nucleotides end up bonding. And that is going to dictate how DNA polymerase runs. So basically on one half of the strand, DNA polymerase can just keep going in one single direction all the way up the entire strand, easy peasy. That's called the leading strand. In the other strand, since those nucleotides are sort of upside down relative to the first strand, the DNA polymerase has to kind of work in sections. And that has to do with the directions of the carbons, and we're not going to get into all that detail of the chemistry behind it, but DNA polymerase can only work in a single direction. And so on one strand, we call it the leading strand because that's the side DNA polymerase is able to just keep going one direction. That's pictured in this picture on the bottom. And then in this picture, the top strand is what we call the lagging strand. It has to get assembled in chunks of fragments. And these fragments are assembled small piece at a time, and that's called an Okazaki fragment. If you see that in a picture or one of the videos, that's what that means. Just DNA polymerase can't operate in any other direction besides the one direction it works. Now, replication is going to be super easy to do a problem for. You just have to know the pairing rule. So if you're given a strand of DNA bases, then you have to be able to insert the correct bases that would pair for it. So if, for example, you were given DNA molecule number 1 here, listed here. If you were given a C, you would have to write in this spot G. If you're given an A, you'd have to write in this spot T. You're just showing how A pairs with T, C pairs with G, and you're just showing the complementary, we call it complementary strand to the strand that they've provided for you. So if you tried to do that again here in molecule 2, and again here in molecule 3, you'd be doing the exact same thing over and over and over again, just writing those same 4 bases in that pairing rule. This is actually a problem from a past Regents exam. So they gave this picture of a DNA molecule with the bases filled in, GC, AT, etc., all the way down. And then they resembled or illustrated replication. So this is the newly replicated molecule. And then this over here on the right is the other newly replicated molecule. And then they asked students, fill in the bubbles. So if you understand what replication is, it's just making a copy. So they literally asked on Regents for students to just copy the letters provided to them. So you don't mess that up. Just make sure you copy carefully. And so you should end up with the exact same sequence of letters as they give you in the first sample DNA to show that you know replication means making the exact same copy of DNA for the 2 new resulting molecules. And we call this semi-conservative because, as you can see here, this side of the first strand is illustrated in black and the right side is illustrated in white. That's supposed to show you that the black strand represents the original half of that original molecule, and then this white strand represents a newly formed molecule from nucleotides that were free floating and then just come in and pair with the newly available base that's available after a helicase opens it up. So you end up just copying the letters, making it look exactly the same as the original. Easy question. So just a couple of questions to review that process if you want to try to answer as we go, like think about what you would answer. Which bonds are broken during DNA replication? So you got to think about what bonds are holding those bases together. What's holding the A with the T? What's holding the G with the C? And we call those bonds hydrogen bonds because DNA is getting split right between those base pairs. What determines the linear sequence of nucleotides in the new strands? Well, if we're talking about replication, then you want your new strands to have the exact same sequence of nucleotides as your original strand. So what determines it? That's the original strand sequences. You want to make a copy of those original sequences of nucleotides. You want your DNA to end up exactly the same as it started when you're doing replication. When replication is complete, how many double-stranded DNA molecules are formed? You start with 1 strand and your goal of replicating it is to end up with 2. How do the 2 new DNA molecules compare to the original one? They should, if everything goes right, be exactly the same. They should be identical. Now, it doesn't always happen. Sometimes things go wrong, but that's a later lesson. The process by which new molecules of DNA are formed are called replication. Okay, so that's my review of replication. I'm going to pop out of my notes for a moment. I'm going to go back to you. I'm going to go back to my participants. Let's see bigger. Where's everybody bigger? That's as big as I can get it now. It's got to get bigger than that. Okay. Do I want to look at that? Okay, so at this time, if you have a question and you want to ask it, you are welcome to unmute yourself and go ahead and chime in with a question. If you don't have microphone available and you want to type it in the chat, there is a chat somewhere. Where is it? There's my chat. If you don't see it right now right away, you could click on more and then chat is there. Select chat. And so if you have a question, you can ask here. Or if you have your mic available, you can just unmute yourself and ask a question. Jewel's raising her hand. That works too. Hi, Jewel.
Yeah, I have a question. When I was doing the Pearson, it said like it switched out the. Oh, wait, never mind. I figured it out.
Not yet?
Yeah.
Okay, that's right. Anyone else? Another question? DNA or replication? I'm going to go into RNA next, and I'm going to go into protein synthesis. So if you have a question on that, just hold. Ethan, yes.
So how does the polymerase helps build like the second half of the DNA strand, right?
Yes.
So like where does it get all the matching bases from?
So you have free-floating nucleotides in your cell, just like you have other free-floating small building blocks of other molecules because you eat them. So when you eat a meal, remember the saying, you are what you eat. You literally build your cells and your body from what you eat. This is true of building your muscles because you eat protein and then your body digests that protein into amino acids, and those amino acids get sent around in your blood and gets to your cells so your cells can use them. You eat starch and that starch is broken down to sugar, and those sugars are going to be sent around through your blood and get to your cells so that your cells can use those sugars for energy. So the same idea is going to happen with DNA molecules that you consume. When you eat the cells of another organism, you're going to be eating their DNA, and you break it down into into nucleotides, the building blocks, and you can reuse those building blocks of nucleic acids to make your own nucleic acids.
Okay.
What's in your cell floating around from digestion end products?
Mhm.
Okay. Any other questions? I don't see many of your faces. Those people are easy to find because they had hands up and faces up. So unmute yourself if you have a question, chime in or type it out. That was the easy stuff, I guess. Okay, so of course, if you come up with something, you're welcome to ask at the end too. So I am gonna switch back to sharing my notes screen. I'm gonna figure out how to get rid of this. I made this too big. There we go, okay, that's better. I'm gonna minimize the chat. I won't see the chat right now while I'm going through the notes. So if you have a question, I'm not gonna notice it there right now. Okay, so now let's talk about protein structure, a little review about proteins themselves, because the whole point of having DNA is to be the instructions for how to build proteins. So we're gonna talk about what proteins there are, why they're important, and how we have a code that builds them. So again, it's gonna boil down to this concept that we call the central dogma. DNA is the code that's gonna code for making RNA, and RNA is gonna bring that code to the mechanism called the ribosome in the cytoplasm that's going to actually put the protein together. And that's got to all happen in a very special sequential order so the protein gets made properly. So that's what we're going to get into right now. So this is a piece of DNA that shows it's kind of opening itself up. It's like unwinding itself. It's like normally twisted and tightly closed, but when those bases need to be revealed for the purpose of expressing a gene, then it opens itself up. So it's opening itself up so that the side that's referred to as the coding strand, in this diagram, it happens to be the bottom half here, that coding strand is now revealed, and that's going to allow a process of transcription to happen. So what you see pictured here is this molecule, that's like an ugly dark green color. This molecule called RNA is forming on this one section of coding strand of DNA. The other side is a non-coding strand. The other side is not going to be copied into RNA at this moment because this does not code for a trait that is immediately trying to be expressed. So this coding strand here at the bottom would represent a gene that is being expressed. So this coding strand is a gene that needs to be expressed. So that sequence of bases, that explicit order of A's, T's, G's, C's, is a very important sequence. They are in a unique order for this gene that's going to be different than another gene, and it's going to be different between different individuals. So this particular order is really unique and important in getting the protein to be built properly. So how do we get this very specific order of bases? It's a code. Think of it that way. It's a code or instructions or a recipe for building proteins, but we can't get DNA out of the nucleus. DNA is too large and too important to leave the nucleus, so it doesn't. What it can do, though, is say, okay, I'm the boss. I have the code here. Here's the code that you need right now. And then in come these little pieces of RNA that come in and match up with DNA following its own pairing rule. So RNA is really similar to DNA, has a lot of similarities, but one key difference when we're talking about pairing, we'll get to that soon, and it's going to form in this process that we call transcription. So DNA opens itself up, allows RNA to form, in a sense, copying its code, but into its own little RNA language, because RNA is able to leave the nucleus. So DNA will close up after this. It's going to not be changed. Nothing is wrong with it. All it did was basically here, take a picture of me. Okay, now I'm done revealing my code. And it goes back to normal. Nothing happens to the DNA. RNA, once this happens, is going to leave and get that message out to the cytoplasm and get to a ribosome, because that's where the code is needed to build a protein, because we know ribosomes build proteins. Right? This is a little bit above our pay grade that we really don't need to know, but it's not as easy as just that one strand of DNA being read. There's going to be some edits that get made when that RNA is forming. So there are sections called introns that get snipped out by special enzymes, and then what's left over are called exons, and they get pieced together to form a more mature piece of messenger RNA that's going to go and deliver that message. A little bit more detail and review about proteins though and why they're important. I'm going to have a lesson for you to do probably next week that's going to involve diving into lots of different types of proteins and why their structure and function are important and what could go wrong if they're not made properly. That'll be leading into mutations next week. So we know muscles. Muscles are proteins. They're structural proteins. We know this picture here, there are proteins in our cell membrane that act as transport proteins or as proteins that detect signals from outside the cell. We know that proteins, another example is an enzyme. Enzymes that speed up chemical reactions, make things move faster. Those are proteins. So we do know quite a few examples of proteins. Here's a few more we know. We know that nerve cells release a protein called a neurotransmitter. We know that cells are able to detect specific signals because they have these surface proteins that can help detect specific shaped molecules like hormones, and many hormones are proteins, and so are the receptors that detect them. This example here is representative of the fact that white blood cells are able to make specific shaped antibodies, which are proteins. Red blood cells are able to make specific shaped hemoglobin. That's another kind of protein. So we know lots of examples of proteins. We make different proteins in all of our cells. Antibodies are made by white blood cells, not by nerve cells. Neurotransmitters are made by nerve cells, not by red blood cells. But what's really fascinating is that all of these cells within one organism, of course, within you, have the exact same set of DNA instructions. So think about it. How is it that these cells have the exact same DNA instructions, barring mutations, which can also happen, but we're not talking about that yet? They have the same exact set of instructions in a nerve cell, in a stomach cell, in a blood cell, in an eyeball cell. But my stomach cell makes hydrochloric acid, not my eyeball cell. My eyeball cell making hydrochloric acid would burn my eyes out, and it would not feel good. How is that possible, that cells with identical DNA function so differently? That is something we're going to get into more in later lessons, but for now, I just want to give you a taste of the idea because it has to do with making the proteins. So you don't make the same proteins in every single cell. You don't make antibodies in your nerve cells. You don't make digestive enzymes in your brain cells. You only make certain proteins by certain cells, even though the instructions are all there. And the simple reason this happens is cells are able to kind of turn off or only express certain parts of DNA code. So remember that part where I said DNA can say, okay, here's the code that you need. I'm opening up so you can see it. That only happens for certain genes in certain cells. They don't all express every single gene. Okay. The other way of saying that is saying that cells can turn on or turn off like a sequence of switches genes so that they don't all get used. They don't all get expressed. More on that concept of expression later on next week. So back to making a protein. Offspring resemble their parents. That's Will Smith and his son. It's kind of an old picture. That's Homer Simpson and Bart Simpson, obviously not actually genetic, it's just cartoon, but I thought it'd be funny. And that is an actress, Reese Witherspoon, and her daughter, and they do look very much alike, as tends to happen. And that is because you inherit the genes for making proteins from your parents. So you make a lot of the same shaped proteins as your parents. So you have more genes in common with your parents, therefore you have more proteins in common with your parents. A little bit more detail about proteins. Hopefully you remember back from biochemistry that a protein is a really big molecule. It's a polymer, and it's made up of its subunit called amino acids. And they have to be in a very specific sequence or order. And I'm going to have a good analogy that'll make that make sense coming up. The sequence or the shape of the protein it ends up twisting and folding into is going to determine its very specific function. You don't have to know the names of these 20 amino acids, but you do need to know that there are 20 amino acids that are present and can be used for building our proteins. You could just see in the picture that they are very different in size, shape, structure, and what you can't see, they're also very different in terms of their biochemistry, their charge, whether they like water or don't like water. We call that being hydrophilic or hydrophobic, their polarity. So based on these characteristics, these amino acids, when they're linked near each other, they might attract each other. Or when they're separated from each other, they might attract each other. Or when they're near each other, they might repel each other and bend and fold opposite directions. So the order we put these amino acids in is going to determine how the protein twists itself and folds itself, behaving in the way that these amino acids do based on their biochemistry. Okay, so let's talk about that sequencing concept. This is a cute picture of a little kitty cat. So I asked my students, how do you spell this picture? It's cat. It's C-A-T. So if I spell C-A-T or just write C-A-T on the board, you're going to put a picture in your mind and you're going to picture a cat. But you can take those same letters and change them around, and you can form other words with those same letters. So I can change C-A-T to A-C-T. If I just put the word ACT on the board, you're not going to think about the cat. You're going to maybe think about the college test that some students take, ACTs, or you might think about the verb of acting, you know, how you act, how someone acts. So we've changed the word by just switching the order. And that idea is going to apply for building proteins as well. So just like the order of letters determines how you say and how you think about a word, that's going to happen when we're building proteins. The order of amino acids that we put together to build a protein is going to determine the shape because of the way they twist and fold, either attracted to each other or repelling from each other, and therefore that's going to determine its function. Remember, we learned enzymes have to be shaped to fit with their substrate. We learned antibodies have to be shaped to fit with their antigens. We learned that neurotransmitters have to be shaped for their receptors. Same thing with hormones. They have to be shaped to fit a receptor molecule. So those specific shapes are dictated by the biochemistry of the proteins that those molecules are made from. That's an important concept. Okay, so as we mentioned earlier, DNA is super important in making proteins. It's the instructions, it's the recipe, it's the code, but DNA is too big to get out of the nucleus. Okay, so what do? Instead, we're going to have the help of a molecule called RNA. Now, there are several types of RNA we're going to get to, but I also want you to know the key distinctions between DNA and RNA, and this you should have notes on already. It was one of the lessons I asked you to run through. Key highlights real quick is that they're both nucleic acids, so they're both in the same macromolecule family. They both have a lot of the same bases, as you can see here. They both have cytosine, they both have guanine, they both have adenine. The one base difference is that you're not going to find any thymine in RNA. Instead, you're going to find this base called uracil. Uracil is going to replace anywhere you're about to put a thymine. Instead, uracil goes there. Biochemically, it replaces thymine. No thymine in RNA. So that's one key difference. Another key difference is going to be the actual R versus the D. RNA stands for ribonucleic acid. So the sugar, ribose, is a little bit different structure of a sugar than DNA sugar. Deoxyribose was DNA sugar. So a little bit different structure of sugar. And then you can see from this picture, RNA is actually only a single strand, whereas DNA, as we learned, is a double-stranded molecule. RNA does sometimes twist and fold itself, so it might not always appear single-stranded, but messenger RNA in particular is going to always show up like this, looking more single-stranded, so you can tell the difference a little more clearly. So that's what the key difference I just talked about. So DNA has the 4 bases A, T, C, and G, whereas RNA has U instead of T. The sugars are different, deoxyribose versus ribose, and then double-stranded versus single-stranded for RNA. Okay. There's only one type of DNA, but there's going to be 3 types of RNA. And I did have you watch, I believe I assigned in Amoeba Sisters about DNA versus RNA. Hopefully that helped clarify those 3 types. We need to know about those 3 types of RNA so that we can further understand this whole concept of the central dogma and how RNA is going to help with the processes of transcription and translation in building a protein. So those are the 2 steps involved in building a protein. So this is another overview of that idea of the central dogma, just a little bit more with location of where those steps happen. So we know DNA is in the nucleus, pictured here, but DNA can't leave the nucleus. So what happens is going to reveal a little section of itself and form a molecule of RNA. And this RNA is specifically a messenger RNA, mRNA molecule. Then that mRNA can leave the nucleus and make its way out to the cytoplasm so that the proteins will get built via ribosome. So a little more detail on that. So if the coding strand of DNA is given to you, like in this picture here, in this slide, if you're given a sequence of bases that is a coding strand of DNA, and you are asked, what would the complementary messenger RNA strand look like? You have to be able to do this concept that we call transcription. So you're going to write out the bases, following the pairing rule, but you're not going to use T's because the rule is there's no T's in RNA. What does RNA have instead? U's. So just like you were doing replication, a C would pair with a G. A G would pair with a C. An A, if you were making DNA, you would write T, but we're not making DNA, we're making RNA. So we're instead going to write U. So I'll just reveal it a section at a time. I'm gonna go through and break it all into threes. So that first set of 3 bases of DNA, which we refer to as a triplet, set of 3, triple, it would code for RNA to form in the sequence GCU, and that would complement DNA's CGA. So we're just going with the pairing rule. Whatever DNA gives us, we're writing out the complementary base, but in RNA language, which means no T's. So the next set of 3, TTG in DNA would code for AAC in messenger RNA. So GAC in DNA would code for CU, no T's, remember, G in RNA. So anywhere you're about to write a T when you're doing these pairs, you just stop yourself and you put a U instead, and you end up with a sequence of messenger RNA. Okay, that's an important rule to remember. And then we're gonna have to take that a step further with the role of messenger RNA. So what we're doing now is we're forming this messenger RNA, and this would be happening in the nucleus because that's where DNA is stored, but DNA can't leave. So the whole point of messenger RNA is to copy DNA's code, but it's copying it into its own RNA language. Then once that process is finished, messenger RNA can leave the nucleus and make its way out and complete the rest of the protein synthesis process with the help from some other molecules we'll get to in a second. Now, when we break down these 3 bases in messenger RNA, we're going to call each one a codon. A codon, this is an important word. A codon is a set of 3 bases that we know, each of these codons, we know which amino acid gets dictated by each of these codons. And we've made these complicated charts and wheels that you're going to have to learn how to read and interpret so that you can figure out which amino acid is brought in by which codon. You'll never have to memorize it. You just have to be able to read the codon wheel or read the codon chart and determine which amino acid is brought in by that. We're going to do that in a few minutes. So what we did here was we formed messenger RNA and we used DNA's code to do that. And this process happens in the nucleus because DNA can't leave, and it's called transcription. So that is transcription, building messenger RNA. That's the first step of protein synthesis. Now, you've got to know about a few different types of RNA to understand the rest of protein synthesis. There are 3 types in total, and the one we mentioned so far was messenger RNA. So key things about messenger RNA is, as we've already mentioned, it forms in the nucleus, and the whole point of it forming is because DNA can't leave. So DNA says, okay, peekaboo, here's my code, and that is going to allow messenger RNA to form, just like you see here in this picture. DNA is unwound in this little area, and in this diagram, the coding strand happens to be pictured on the top. Okay, so in this diagram, the coding strand is this top strand, and I know that because that's the strand that has mRNA forming on it. The bottom strand here has no mRNA forming, so I know this is the non-coding strand. The enzyme that's involved with building RNA is almost the same as the enzyme involved with building DNA. It's polymerase, only this time it's called RNA polymerase. So RNA polymerase helps with building messenger RNA during the nuclear process of transcription, transcribing DNA's code into RNA form. The goal of making this type of molecule, making mRNA, is because it is able to get out of the nucleus. There are little holes in the nuclear membrane called pores, and messenger RNA is small enough to get through those pores and make its way out to the cytoplasm and eventually find a ribosome so that it can be read for the concept of protein synthesis. Now, each set of 3 bases called a codon is going to be an important concept because that RNA is going to be read in 3s. It's going to be read 3 sets of bases at a time, and that will be what dictates which amino acid gets coded for. Okay, the next type of RNA, remember I said there's 3 RNAs. The first one was messenger RNA. This one is called ribosomal RNA. all you really need to know about this one is that it literally builds the ribosome. That's it. So it's a component of building a ribosome. So a ribosome typically comes in two halves. There's this large orange chunk here, that's called the large subunit, and this smaller chunk here, pictured in like a lightish bluish color, is the small subunit. And these pieces assemble around a messenger RNA when they find it in the nucleus, and then they read and translate that messenger RNA and put together the protein by putting one little amino acid together at a time. So you see this growing chain of little blue beads. That's supposed to represent the amino acids each getting put together as this ribosome is reading this sequence of messenger RNA. So ribosomal RNA, you're not going to see bases for it. You're not going to interact with it in any way in problem solving. You just kind of got to know it's one of the RNAs and it builds a ribosome. That's why it's called ribosomal RNA. It literally forms the ribosome. The third type of RNA is going to have a little more involvement in the process of building proteins. So this one is called tRNA, and the T stands for transfer RNA. So its job is going to be to transfer the building blocks of proteins. Those are amino acids. It's going to transfer those amino acids to the site of where they're being built at the ribosome. And it's going to bring the right amino acid at the very right time, the very right moment when that protein is being built. How does it know what the right amino acid is going to be? It's going to know, I'm getting a chat message here. Do the back, okay, just mess around with backgrounds, guys? Okay, don't distract me. All right, going back. Thought somebody had a question. Okay. So sorry, so transfer RNA carries the amino acid to the ribosome, so you get the right amino acid in the right order at the exact right time. So if you want to spell cat, you got to put the C first, and then the A, and then the T. If you put the T first, you don't have cat anymore. So it's the same idea when you're building a protein. If that protein is meant to have the amino acid tryptophan before the amino acid alanine, then it's got to be brought in the exact right sequence. Okay, and that happens by tRNA bringing the amino acid over. So you might see tRNA pictured in a few different ways. This picture here on the left is a two-dimensional diagram of tRNA. It tends to kind of fold over itself. It is single-stranded, but then it folds over itself, so it kind of appears double-stranded, almost like a DNA. There's a little sticky attachment point here at the top where an amino acid will bind. Here at this top half of the molecule. And then at the bottom half here of the molecule, you see a set of 3 bases that are poking outward. And that set of 3 bases is going to be important. It's called an anticodon. And that is going to be the set of 3 bases that matches up and complements a codon. Remember, codon was a set of 3 bases in the other type of RNA we called messenger RNA. So messenger RNA is going to have a set of 3 bases that it made by copying DNA's code, and it's going to allow the right amino acid to come in by having the correct transfer RNA molecule anticodon match with it. We'll get some practice with this. There's more 3-dimensional diagrams of tRNA. It's kind of T-shaped, so that might help you realize and recognize it's a transfer RNA. But if you see an amino acid attached to it, then you're going to know it is a transfer RNA. And its sequence of 3 bases we call anticodons because it's going to follow that pairing rule and match up with the codons of mRNA. So here's some practice questions about RNA, just like making sure you know about the different types of RNA. And we're going to do these, and then I am going to go through more detail on protein synthesis. Okay, so I'm going to leave these up. And I'm going to have you look at them and just try your best to answer them, maybe like on the side of your paper or wherever you can jot it down, and we'll go over them together. I'm going to also say if you have a question now, please unmute yourself and ask a question. If you have no question, just think about those questions. Did I just take away the questions? You can't see it anymore? Hold. I was trying to look at my chat. Can I look at my chat at the same time as showing you my screen? It does not appear that I can. All right, so if you have a question, unmute yourself. Wait, who's writing on my screen? I imagine you think you're writing on your screen, but you're somehow writing on my screen. Thank you. I'm gonna give you a minute to think about them and then we'll go over them.
Can the RNA be like reused?
Hello, who's talking?
Casey.
Hi, Casey, what's your question?
Like, is the RNA like reused when like during the transfer, or is it like made anytime it needs to like?
A new one is made every time a gene needs to be expressed. But then we do break it apart and reuse its parts. So those RNA nucleotides do get reused within our cells.
Okay.
We don't waste stuff. So let's look at these questions together. Number one, in the synthesis of proteins, what is the function of messenger RNA? Messenger RNA is going to be the one that is able to copy DNA's code so it can get out of the nucleus. So that's going to be best said by choice 2, carrying information that determines the sequence of amino acids. The information that determines the sequence of amino acids is the DNA sequence of bases. So if DNA reads T, 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 but that code of T's can't get out of the nucleus because DNA is too big, then messenger RNA is going to come in and form A, copying that code into its own language so that it can eventually relay that to get the amino acids to be built properly. The sequence of 3 bases in a messenger RNA, this is number 2, is called a codon. Which base is normally used in the synthesis of RNA but not in DNA? You've got to know that rule. RNA has uracil, that's choice 2. If a portion of a messenger RNA molecule contains a given sequence, if you're given AAU for a messenger RNA, then you'll have to correspond and write out the transfer RNA base sequence. So if messenger RNA reads AAU, then the RNA of transfer RNA is going to complement it with that same pairing rule. And still remember, no T's allowed because we're making another kind of RNA. Transfer RNA is going to have U's just like messenger RNA. So AAU would pair up and complement with UUA, which is choice 4. Number 5, the code of a gene is delivered to the enzyme producing portion of the cell. Forgive me, mine is cut off a little bit by my controls. Let me move it over. By a hormone, nerve impulse, messenger RNA molecule, or DNA molecule. The code is delivered to the portion of the enzyme producing region. The DNA can't deliver the code. The delivery messenger is the messenger RNA molecule. So 5 is going to be choice 3. And then number 6, which statement best describes the relationship between cells, DNA, and proteins? So this is that concept of the central dogma, the idea that DNA is the code for building proteins. So cells contain DNA, and DNA is the code for building proteins. And the best statement that says that is choice 1. Right? Some of these will maybe make more sense even after I show you the details of protein synthesis. I know you read about it, and I did assign you a Pearson on it. But I do want to go through it with you also. So let's do that next. Okay, so you're going to have to understand how those codons, remember a codon is messenger RNA, but in 3 bases at a time, how those codons translate into which amino acid gets put in the protein. The protein has to have the correct amino acid in the right time, in the right sequence building it. So we know when we figured out which codons code for which amino acids. So we have this chart here that you're going to have to learn how to use. And we also see this in a wheel form. Your textbook uses a wheel form as well. And I'll show you that on another slide coming up. So the codon for tryptophan, you don't have to know it, but you have to find it. So tryptophan is an amino acid. It's associated commonly with turkey, and when you eat a lot on Thanksgiving, it's associated with falling asleep and being sleepy because you're taking a lot of tryptophan. there's some fallacy to that statement, but maybe you've heard of tryptophan for that reason. So find tryptophan on your codon chart, and if you can see my mouse highlighting it, it's going to be right here in this second column. There it is. Yep, whoever's writing on theirs is writing on mine too. You got to teach me how to do it. Ethan, is that you? Are you writing on it? You're
No, it's
muted.
not me.
Oh, yeah, Ethan, was that you?
No, it wasn't me.
No, it wasn't you? Oh, some more of you know how to do Zoom more than I did. See, if I just click, it's going to just advance to the next part of the slide. So if you find tryptophan here on the chart and you're able to read the chart properly, you're going to see the left side of the chart tells you the first of the 3 bases. The top of the chart tells you the second of the 3 bases. And the right side of the chart tells you the third of the 3 bases. So if you go ahead and highlight or find tryptophan, you're going to find that to get tryptophan, you need the codon UGG to get tryptophan. Next problem, for leucine, there are how many different codons? So for that, we have to just look around the chart and say, where's leucine? I see some leucine down here. And where else is leucine? Oh, there's 2 more leucines right over here. So basically, if I see leucine listed 1, 2, 3, 4, 5, 6 times on this chart, that means there are 6 different codons to get leucine. This is going to make sense and be important later when we're determining if changes in the DNA are going to have an impact on the amino acid sequence. So what this shows me is that there's 6 different ways to get the amino acid leucine. So if one base happens to change, you might still get the amino acid leucine like you're supposed to, if you're supposed to. So there's a few different ways to get some of these amino acids. Remember, there's only 20 amino acids, but there's 64 codon combinations. So of course, there's going to be some duplicates. So the codon, number 3, the codon for GAU is 4. So you just go G is the first one here on the left. A is the second base up at the top. And so it's got to be somewhere in this row, but in this column. And then on the right, it tells me U is this one right here. So GAU has got a code for aspartic acid. A stop codon. Okay, so sometimes the chart will tell you start, stop, and sometimes the chart will tell you the amino acid that is the start or stop amino acid. So don't get confused if you see it 2 different ways. So in this chart, it's telling you which codons code for stop. That's basically a signal to the ribosome saying, okay, you're done building the protein, stop. This is the end of the line. That's it. So a stop codon that has the second base that is G. The first and third bases must be. Okay, so we need a second base that's G. That means it's got to be somewhere in this column here with the second base G. And then it's got a stop right here in this spot. So figure out which combination of letters gets this stop right here. So it's going to be U, G, A. So U and A are the first and third bases to get that codon. If this is a little tricky, there is Amoeba Sisters on how to read a codon chart that I do plan on assigning you later this week, I think, just to get you a little bit more practice. Now, not all codon charts work in that manner. Sometimes you're going to see the mRNA codons presented in this circular manner, in this wheel. And the wheel is just as easy to use as the chart. Some people have a preference one way or the other, that's personal preference, but you have to be able to use whatever's presented to you because you're not memorizing any of these charts. You're just interpreting them. So if you're given a codon and you have to find it on the wheel, you need to start in the center. So if I give you a random codon, I'm going to say GGG, G for Gina, G, G, G. If you start in the middle and work your way out, you'll see that GGG leads you to this amino acid here called glycine. If we pick CCC, start in the middle, CCC, work your way out to the next C, work your way out to the next C, and you're going to find the amino acid proline. Sometimes there'll be abbreviations for these amino acids. It might just say pro, or it might just say gly, G-L-Y for glycine. That's not something you're ever going to have to memorize. You will just have to be able to interpret that by starting in the center and working your way out. So you can see the stop codon is over here on the wheel, stop. So UAA is a stop codon, and so is UAG. They're both stop codons because the A and the G are both here in this little wedge of the wheel, and both lead to that stop codon. Another stop codon over here, UGA. Oops, clicked too fast. Okay, so this is just another way of interpreting mRNA codons. mRNA codons are the sequence of 3 bases at a time that are read by a ribosome to determine what the next amino acid is going to be in the sequence. Okay, so a little more detail on this. Transcription is that step we've already talked about where we make mRNA from DNA in the nucleus. So DNA is going to open itself up and say, peekaboo, here's my code, this is the gene you need, and then whatever that gene reads in DNA language, messenger RNA comes and forms in messenger RNA language. So this is happening in the nucleus. It involves RNA polymerase forming mRNA. I hope you're not trying to write this down right now. You should have had it written down already, and if you didn't, you'll do it after the fact. But what's important is that mRNA can then leave the nucleus once it's done, once transcription's finished. We've made mRNA so it can leave the nucleus and move into step 2, which is what we call translation. Don't get confused, transcription with translation. Transcription first, translation second. Translation is just the building of the protein. You're translating messenger RNA's code into the correct sequence of amino acids. And I have a good animation that makes this make sense. So you should have these notes already jotted down because that was one of your assignments this week, was to look at my lesson 5 on the website and write down this information. So you should know this is happening at the ribosome because that's where proteins are built. And it's going to involve tRNA bringing the correct amino acid to the ribosome at just the right time based on what mRNA codons read. So tRNA has an anticodon that's going to match up with mRNA's codon, and so that's how we're going to know this is the right amino acid that we need at this exact time. I think this simulation is going to make it easier for you to understand because it's going to show it kind of in motion, but I can stop it and do it step by step. Here's what I held while you were on full screen mode. That's my mail. Never mind, ignore my mail. So we are now going to look at this simulation.
We're gonna ride it by Lee and Lainey in a bit.
Whoa, was that someone's little brother or sister? Hello? Okay. Okay. It says my screen sharing is paused. I don't know why it says that. Can you guys see my screen? Stop video. No. Resume share. Okay. Get rid of that. Okay. All right. It says it resumes share. No, resume share. There we go. But I don't want my PowerPoint anymore. I want this website. Sorry, technical difficulties. Trying to figure this out. Okay, I need someone to unmute themselves and tell me what you see on my screen, please.
What?
Hi, can you see my screen?
Yeah.
Okay, what do you see?
Protein synthesis simulation, step by step. And then, like, to the left, there's a bunch of little slides.
Okay, so that's not what I want you to see. So you're not seeing my actual screen.
Oh, yeah, and I think I could draw.
Well, all right, I'm gonna close that slideshow. Want to save your Zoom? No, wait, what?
Oh, I see that.
Okay. You see that it's asking me if I want to save?
Yeah.
Okay, I'm gonna save that because that's not what I want to show you anymore. But I want to show you something on a browser. Oh, screen sharing has stopped as the shared window is closed. Okay, so, but now I see all of your faces. Okay, so now I'm gonna hit share screen again and hopefully I can pull up that browser. Okay, that's the problem. Share. How about now? Do you see a picture of cells?
Yes.
Yes, okay, wonderful. Let me make this bigger. Thank you for your patience. Okay. Okay, so what we're looking at here is a bunch of cells, and once I hit the animation, it's gonna let us zoom in to the nucleus where we'll see DNA, and we're gonna see the DNA get transcribed to messenger RNA, then we're gonna see the messenger RNA go to the ribosome to be translated by the ribosome, and we're going to see the correct transfer RNA molecules come on over and bring the amino acids. So we're going to do it step by step. So first step, we're zooming into the nucleus. There's chromatin. We're going to zoom into one strand of chromatin and look more closely. It's really just a twisted, coiled up piece of DNA. Now that DNA has to, that's an enzyme that's going to attach. It's going to help open up the DNA. And now, there we go. We've seen the DNA open up. Now, it's already been revealed which strand, I just pause it for a second, the strand that is going to be the coding strand in this simulation is this bottom strand. The top strand is not going to be copied. So what we're going to see happen is individual little free-floating nucleotides of RNA will be coming in to pair up with these newly separated pieces of DNA. So if I backtrack a little bit, okay, see, this is DNA. Those are the bonded base pairs of DNA. But the next step is going to be to open up that DNA and break those hydrogen bonds between the bases. Boom, they're broken. And now we're going to see individual RNA nucleotides come in and bind, following the pairing rule. But again, remember, you're making RNA. You're not making DNA, so you're not going to have any T's, no T's in this whole process. So we keep going, keep going. We're making a big, long RNA, messenger RNA molecule. Okay, see that pairing is always consistent, G with C, U with A. And when we get to the end of our line, the end of the gene that needs to be transcribed, that is going to be where it detaches. Okay, next step. So it's going to detach itself. The DNA, oh, I jumped too far. Start, continue. Okay, the next step is, okay, that enzyme detaches, that RNA is going to detach, the DNA is left alone, nothing changed about that DNA. Now this RNA is able to go and leave the nucleus and make its way out to a ribosome. That chunky brownish thing that just formed around it is a ribosome, and I want you to see what's happening. You see a little set of amino acids, these little brown spheres here at the top. They're going to form with every one of these little green guys coming over. These green guys are transfer RNA molecules bringing the amino acid. And the correct amino acid is being brought because these bases on the tRNA match these bases on the mRNA. So if we look at it step by step, the next tRNA has to read CCG to match up with this GGC. Okay, step by step, the next one also has to match, the next one also has to match, and these tRNAs have a certain amino acid depending on which anticodon base sequence they have. Okay, that's how that gets determined. Oh, I hit the wrong button, my bad. Translate. Continue. Getting back to where we were. Okay, oh, too far back. Okay, one step at a time. So there's a tRNA coming in. There's a tRNA coming in. The amino acids join together. They're actually forming peptide bonds, holding those amino acids together. And then once we get a long enough chain of amino acids, we're going to see those amino acids start to either attract each other or repel each other based on their own biochemistry. So maybe it looks like Met and Lou seem to like each other and they're not being repelled. They're actually kind of folding so that they're near each other. And it would really be much longer than this. You can have thousands of amino acids to make up one protein. This is obviously just a simulation. But this protein is now folding in a very unique way depending upon the order of amino acids that it contains. And if I wanted to, I could go in and edit the DNA sequence from the program, and it would have to build me a different protein based on the DNA sequence that I put in. And that is that. Okay, so I want to go back to looking at you and see if you have any questions for me. Let's pull that back up. Go back to my participants. Where are you guys? Where's your pictures? There you are. Okay, those of you who are showing me. Okay, Naresh, is that Vin Diesel? Who's in your background there? I can't tell. It's some bald guy, but you're blocking him. I can't tell who it is. Okay, who has a question? I know that was really long and really complicated. If you made it all the way through, you are a hero. I love you. Good job. I promise you it wouldn't have been that long and intense in one period. It would have been like 3 or 4 periods of going through all that detail, but you did so much pre-work. I thought we could get through a lot of it all at once. So thank you. I appreciate it. Unmute yourself if you have a question. I see, what is a promoter? I don't know how long ago that question was asked, but we were talking about a promoter when we were talking about where the enzymes start their action of replication. So that was like the first half of the slides where we were talking about how DNA knows where DNA polymerase knows where to anchor itself in order to start copying and making new strands. Oh, Naresh, now Michael is your background? Come on, you're just gonna confuse people.
Yo, Naresh, yo, come on.
Not the best picture.
It was
Keep it with Kermit the Frog, that's cuter. No offense, Michael. It wasn't your best side.
I'm very offended. I'm good.
Okay, so a promoter is just it's a place made out of typically it's like the enzyme. It's the enzyme that lays itself. That's going to be like it's a region of RNA or a region of DNA. That's like a starting point, and it's read by certain enzymes to dictate to the building enzymes where to start. That's the important part of a promoter. But honestly, it's not really an important part for us. So transcription, translation, are we good? 2 main steps of photosynthesis. Protein synthesis. If you're going to build a protein, first thing you need to do is transcribe the DNA into messenger RNA. Second thing you need to do is translate that RNA into the actual pieces of the protein, into the amino acids that make it. I'm going to send you more practice. You're going to do another web quest. I'm going to see, I think I have another simulation that's similar to the one I was showing you, but you can activate and you can move through. So it might be a little bit more memorable to you to do it as well. I'm going to get you that one as well. Any other questions before we wrap it up? You can type it or you can unmute yourself and just ask. Naresh, you're making me dizzy. No, I don't hear anyone unmuting and I don't see anyone typing. You guys, did you all find where you could type? Michael, you are very into this distance learning thing. You've got your headset. I'm guessing you use that for video games.
Yeah, I'm just on my computer.
Oh, Naresh has it too. Headset, ready to roll. I'm not that high tech. No? Okay. All right. Well, then, if you have no other questions for me, if you do have a question for me, of course, you can. Oh, Lenny's joining in, showing off his fancy headset too. Hi, Lenny. I see you. And the lovely Golden Gate Bridge.
Not the Golden Gate.
Now, I missed you guys. I'm happy that you're showing me your videos. That's very nice. Thank you. I'm really happy to see you. I hope I get to see you on Monday. We're supposed to go back to school, but I don't know. Everybody's telling me different things. We'll see what officially comes out. If you need me, you know where to reach me. Message me, email me. Try to keep it within normal-ish school hours. Do the ed puzzles. They are useful. If you do a bad job at them, text me and I'll reset it for you and you could try it again. I really think that's one of the greatest tools about this distance learning thing that I've discovered is those ed puzzles. So I'm going to keep assigning them because I think they're useful. All right, no other questions? Nothing being typed? No one unmuting themselves? All right, well then I'm going to sign off. I'm going to hope this recorded and see if we did an okay job getting this out to anyone who couldn't join us. I appreciate your time today, guys. Thank you so much. If we do this again, I don't think it would be this long. We did a lot and it was confusing stuff, so I tried to go in a lot of detail. So hopefully next time we do it, it won't be that long. Thank you. I'm not sure how to stop the recording.
Thank you.
Oh, there it is. Okay. All right. I'll miss you. I'll talk to you soon.
Do you see my drawings?
We'll be in touch. What?
You see my drawings?
Where's drawings?
Oh, the smiley face.
Oh, like the little purple smiley face in the corner? That was
No,
you?
there's like a big smiley face.
No.
It's
No,
not
I don't
showing
see
up.
that. I don't see that. I see everyone's picture. No, I don't see any smiley face. Sorry.
No.
Maybe next time.
No.
As long as you're not drawing on top of something I'm teaching. 'Cause then I kick you out because I could do that.
It's getting erased.
Bye. That's not me. Someone else must be erasing
Bye.
you.